photosynthesis. Now, as we know that it is basically a process, we can even say that it is a physico-chemical process. It is a physico-chemical process in which green plants also known as autotrophs in the presence of sunlight with the help of chlorophyll prepares their own food. So this is basically what photosynthesis is. So it is a physico-chemical process. We can say that it's a process by which green plants, we even call them as autotrophs, since they can prepare their own food. That's why they are known as autotrophs. So we can say that green plants, in the presence of sunlight with the help of chlorophyll, basically they prepare their own food. Now we can say that all animals, directly or indirectly, okay, we can say that all animals, or even we can say all organisms, directly or indirectly depends on green plants for their food. We know that. And these autotrophs, or we can even say green plants, they can synthesize their own food. Now we have already discussed that these green plants, they utilizes basically sunlight and chlorophyll. Besides sunlight, chlorophyll, we can say that they also utilizes carbon dioxide and water. Just a second. Okay, so these are the components that are used during photosynthesis. We can say that these are the components that are used during photosynthesis. In this process, basically sunlight is the major source of energy. Sunlight is the major source of energy. Now chlorophyll is basically the pigment. These are pigments that absorb sunlight, Now this carbon dioxide, it is basically we can say that its main source is atmosphere. It is a raw material that is used during the process of photosynthesis. And water, we can say that plants generally absorb capillary water. They draw water from the environment. They utilizes capillary water. Okay. Now, we'll find that several experiments were conducted by several scientists to demonstrate the process of photosynthesis. Okay, so let us discuss those experiments that were conducted in favor of photosynthesis. Now, basically, in your previous class, you might have read about the experiment, just the experiment where presence of starch is generally observed. I hope you have already discussed that experiment, presence of starch. Now, let me tell you a bit about this experiment. Name of the experiment is presence of starch. Now, in this experiment, generally what is done, a variegated leaf is generally taken. Okay, variegated leaf. Now, what is meant by variegated leaf? Variegated leaf means a leaf, with a portion of leaf is chlorophyll-less. A variegated leaf means a portion of leaf is without chlorophyll. Is without chlorophyll. ফর এক্সাম্পল একটু আলাদা করে বলে দিচ্ছি, সাপোজ একটা এরকম লিফ আছে, তো তার কিছু পার্ট লেট ইট বি ক্লোরোফিললেস, let it be chlorophyll less. So this kind of leaf is basically known as variegated leaf. Now the other portion, অন্যান্য যে পোর্শনগুলো আমরা দেখতে পাচ্ছি, সেই পোর্শনগুলোতে ক্লোরোফিল রয়েছে। তো কিছু পার্টে ক্লোরোফিল থাকে, কিছু পার্টে ক্লোরোফিল থাকে না। তো some parts are chlorophyll less and some parts are we'll find that chlorophyll is present. So that kind of leaf is generally known as variegated leaf. So basically we'll see that a variegated leaf is taken, or we can even, if a variegated leaf is not found, then in that case what we generally do, we cover a portion of the leaf with black paper. We cover a portion of leaf with black paper, and generally what is done, it is then kept in sunlight. And presence of test for presence of starch is conducted. Test for presence of starch is conducted, and what is generally observed? It is observed that this chlorophyll-less portion do not show presence of starch. Chlorophyll-less region do not response to starch test. On the other hand, the region with chlorophyll basically it provides response to starch test. We can say, it can be concluded that this region shows shows the presence of starch. It responds to starch test. Next one is an experiment conducted by Joseph. experiment conducted by Joseph Priestley. Now what he did, now he took a closed bell jar. Okay, he generally took a closed bell jar, and within the closed bell jar a burning candle was kept. Burning candle, and along with that a rat was also kept within that closed bell jar. Now it was obvious. It was obvious that basically what was observed, we can say that the rat died. Okay. Now what he did? He again took a closed bell jar. Within the bell jar, he generally kept a burning candle and a rat, and besides a rat, a potted plant. A potted plant was kept. A potted plant was generally kept. Okay. Now what was observed? Now what was the observation? It was observed that the rat lived, or we can say that the rat survived. And in this case, what was the observation? The rat died. The rat died. So, what Joseph Priestley concluded? What he concluded? What was the conclusion? He believed that basically the foul substances released by burning of candle, in this case he concluded that the rat died due to the release of foul substances by burning candle. So in this case it was concluded that a rat generally died due to the release of foul substances by the burning candle. Now in this case it was observed that the rat generally survived, that the rat lived. So basically it was concluded that the foul substances released by the burning candle was removed by the pure substance, released by the plant. Now later on it was discovered that this pure substance, later on it was found, or we can say that Joseph Priestley in 1774 he discovered oxygen, isn't it? So basically this pure substance was later on found that it was generally what? It was oxygen that allowed the rat to live. Is it clear?
Yes, sir.
Okay. Now, another person whose name was Jan Ingenhousz. Uh, Actually, I don't have any idea how you are going to pronounce it. I have made my own pronunciation, so pronunciation is really not the factor. Jan Ingenhouse, whatever it is. So this person, what he basically did, what he concluded. He concluded that in this experiment, the burning candle is very much essential. It is also an important point to note down. No one, or we can even say that basically Joseph Priestley never made this conclusion that this burning candle, it is very essential, is essential. This burning candle is very much essential for, we can say, for this experiment. Okay, it is very much essential for this experiment. Anyway, why it is so? Because this burning candle, generally we know now. What we can say that burning candle, it generally emits carbon dioxide, and this carbon dioxide is utilized by plant. Why it was said that it was essential? Because the foul substance emitted by this burning candle was utilized by plants. So as a result, the plant releases oxygen or this pure substance. Okay. Joseph Ingenhouse, he also performed several other experiments. Let us find out that. What he did, we can say that he performed a series of several experiments. In one such experiment, he observed that generally when an aquatic plant, it is kept in bright light, when it is exposed to bright light, small bubbles are found around the plant. Small bubbles are found around the plants. In contrast, we can say that when these aquatic plants are kept in dark lights, basically we can say that bubbles were not seen, bubbles were not observed around the plants. Now, why these bubbles were released? It is because these bubbles were, it indicates the release of oxygen. So in bright light or in presence of light, basically the plant performs photosynthesis, and if we look at the chemical reaction of photosynthesis, then we'll find that oxygen is given out during this process. So this experiments generally proves that, and not only this experiment, the experiment that we have discussed earlier, or basically before this, generally these experiments proves that photosynthesis, it helps in release of oxygen. Now, in your introduction only, it is generally written that there are two main importance of photosynthesis. Two main importance. Now, what are those importance? Basically, importance of photosynthesis. There are several such importance. তার মধ্যে দুটো হচ্ছে খুবই ইম্পর্টেন্ট, আমরা ইম্পর্টেন্স দেখতে পাই। এক, it synthesizes food, and the other one is that it helps in release of oxygen, which is very much required for each and every living organism, basically each and every living organism, even plants as well as animals, for their survival. Oxygen. Is it clear? Now, another person whose name is T.W. Engelmann. Okay, Engelmann, or whatever you may pronounce, Engelmann. Now, he also performed an experiment. Let us find out, or let us discuss that experiment. Now, experiment by T.W. Engelmann. Okay, now what he did, he split white light through a prism. Now what will happen if a white light is splitted through a prism? Now what will happen? Can you tell me please?
The colors will spread into 7 colors.
How many?
7 colors.
7 colors. So it is due to, why it is going to split? It splits according to their wavelength. And what we generally find is that a VIVGEOR is formed. Basically, what we'll find that when a cladophora, a green algae, it is generally exposed to this VIVGEOR. Now this cladophora is taken in a suspension of aerobic bacteria. Now, what is meant by aerobic bacteria? Bacteria that utilizes oxygen. So basically, যখন photosynthesis হবে, cladophora is a green algae, it is going to perform photosynthesis, or it is going to synthesize food with the help of photosynthesis. So basically, যখন photosynthesis হবে, evolution of oxygen is going to take place, means oxygen will be evolved. So obviously these bacteria, since they utilizes oxygen, we can simply say that they will be found in the area where more and more oxygen will be evolved. Is it clear? ভেবে দেখো, এরা oxygen utilize করে. Cladophora is generally taken in a suspension of aerobic bacteria. মানে aerobic bacteriaর সাথে cladophora নেওয়া হলো, একটা ক্ল্যাডোফোরার একটা সাসপেনশন তৈরি করা হলো। সাসপেনশনটা কিসের ছিল না, এরোবিক ব্যাকটেরিয়া। সো অবভিয়াসলি ক্ল্যাডোফোরা it is going to perform photosynthesis, and due to photosynthesis oxygen will be evolved, and since this bacteria utilizes oxygen, so these bacteria will be found more and more in the region or in the area. বলতে লাইটের কথা বলা হয়েছে, in the region or the area where more and more oxygen will be evolved. Is it clear? এটা বোঝা গেছে, প্রিন্সিপালটা বোঝা গেছে? Okay. So what we'll find, these bacteria were usually they were used to detect evolution of oxygen. Now, so basically what was observed? Bacteria accumulated observation. Now it was observed that bacteria, these bacteria, they accumulated in the region of dark, we can say blue and red light, sorry, in the region of blue and red light. Now what it can be concluded? What will be the conclusion? Yes, can you derive the conclusion? Blue and red light এ সবথেকে বেশি ফটোসিনথেসিস হচ্ছে বলে সবথেকে বেশি অক্সিজেন জেনারেট হচ্ছে, আর সেই রিজিয়নে সবথেকে বেশি ব্যাকটেরিয়া অ্যাকুমুলেট হয়েছে, কি বলতে পারি না?
হ্যাঁ।
So it can be concluded that photosynthesis, rate of photosynthesis, rate of photosynthesis is higher in blue and red region of the spectrum. We call this as spectrum, isn't it? Blue and red region of spectrum. Okay, is it clear now?
Yes, sir.
Okay. Now, as you know, it was already discussed that several experiments were performed. So এতক্ষণ যেগুলো দেখলাম ভেবে দেখবে যে first one, it helps to detect the presence of starch, or which part of the what we have discussed is that generally the portion with chlorophyll, they show photosynthesis, so chlorophyll is very important for chlorophyll, এটা দেখেছি। তারপরে যেগুলো দেখলাম that Jan engine house experiment, সেখানে আমরা দেখলাম যে evolution of oxygen occurs due to photosynthesis. Okay, Joseph Priestley's experiment থেকেও এটা বুঝেছি। এঙ্গেলম্যানের এক্সপেরিমেন্ট থেকে কি বুঝতে পারছি? এঙ্গেলম্যানের এক্সপেরিমেন্ট থেকে বুঝতে পারছি যে বেসিক্যালি কোন রিজিয়ন, স্পেকট্রামের রিজিয়নে সবথেকে বেশি ভালো ফটোসিনথেসিস হয়, মানে স্পেকট্রামের কোন রিজিয়নে রেট অফ ফটোসিনথেসিস সবথেকে হাই হয়, তো সেটা বুঝলাম। Several other person also conducted several other experiments. For example, Van Leeuw, okay, he conducted several experiments and he basically concluded several statements. Let us find out that. Van Leeuw basically he concluded that photosynthesis is a light dependent, just a minute, running out of ink, light dependent process. Okay, and in this process we can say that hydrogen form a suitable oxidizable compound, and in this process basically what is the role of hydrogen? Hydrogen form a suitable oxidizable compound. This is very important, you can mark it, this is very important. Hydrogen form a suitable oxidizable compound, and basically we can say that it reduces this hydrogen. It is an oxidizable compound, so we can say it reduces carbon dioxide. It reduces carbon dioxide. Now the source of this hydrogen is H2. Source of hydrogen is basically, we can say, is H2. So basically he performed an experiment. We can say that he took green purple sulfur bacteria. Okay, what he did, green purple, green sulfur and purple sulfur, not green purple. You can write it down if you are taking a note. He took green sulfur and purple sulfur ব্যাকটেরিয়া। তো কি করলো, গ্রিন সালফার এবং পার্পল সালফার ব্যাকটেরিয়া নিল। নাও বেসিক্যালি when H2S was used instead of H2O, he took this bacteria for his experiment. He used H2S. Now H2S is actually it is going to act as a source of hydrogen. He took H2S instead of H2O. Okay, so basically we will find that sulfate is released. We can say that sulfate is released, and when H2O will be taken, oxygen will be released. So photosynthesis আমরা দেখতে পাই যে অক্সিজেন রিলিজ হয়। So what is the source of this oxygen? অক্সিজেনটা আসে কোথায় থেকে? H2O থেকে। এই এক্সপেরিমেন্টটা থেকে এটাই বোঝা যাচ্ছে যে instead of H2O, when H2S is taken, so basically sulfate is released. So obviously this water, when it is utilized in the process of photosynthesis, basically what we find is that oxygen is released, and this water, it also act as a source of hydrogen, which we can say it's a form of suitable oxidizable compound that reduces carbon dioxide. So photosynthesis, we can simply say that photosynthesis is a oxidation reduction reaction. Oxidation reduction reaction. Okay, now where does this photosynthesis takes place? Where does photosynthesis takes place? We can simply say that it takes place in leaves. In green leaves. কিন্তু একটু মাথায় রাখবো, আমাদের হয়তো it is not written or it is not given in NCERT, but we should also remember that green parts of plants, green parts of plants, সেখানেও photosynthesis হয়. So we should remember this. But green parts বলতে basically we'll find a stem, even flower. Calyx is a basically portion of flower. It's a part of flower. Sorry, calyx. It's a part of a flower. So we can even say calyx. Generally, they are green in color. So since they are green in color, so we can say that photosynthesis takes place within the calyx of flower also. So it's a parts of flower. So different part, green part আমরা বলতে পারি যে এই photosynthesis হয়। But in NCERT only যেটা আমাদের দেওয়া আছে সেটাই ডিসকাস করছি, যে বেসিক্যালি it occurs in green leaves. It generally occurs in green leaves. Green leaves আমরা দেখো mesophyll cells. Mesophyll cells. Now what is meant by mesophyll cells? Now these are parenchymatous cells. Parenchymal cells with chlorophyll. Mesophyll cells are those cells which are actually parenchymal cells with chlorophyll. Sorry, with chloroplast. Now chloroplast are the organelles found within the plant cell. Now, if we discuss the structure of a chloroplast, then what we'll find that it is a double membrane bound organelle. It's a double membrane bound organelle. The outer covering is known as outer membrane. The outer membrane covers the inner membrane. Generally, the inner membrane, it encloses the stroma. Stroma, or we even call it as matrix. It's a jelly-like substance. It's a jelly-like substance. Now, within this stroma, what we generally find is, bag-like structures. Now, this bag-like structure, each bag-like structure, they are known as thylakoid. Thylakoid. Now, these thylakoid, they are stacked upon each other. যেমন যদি কোনো ওই 5 টাকার বিস্কিটের যখন প্যাকেটগুলো নেবে, you will find that these those biscuits are generally stacked upon each other. They are placed in such a way that it appears that they are stacked upon each other. So similarly, এখানে thylakoid, it forms similar kind of arrangement, and this arrangement of thylakoid is known as grana. So this is the difference between grana and thylakoid. Grana is made up of several thylakoids, and each bag-like structure is known as thylakoid. Now, what we'll find is that this grana, or even we can say that these thylakoids, they are form an interconnection. We can say that these grana are interconnected. They are generally interconnected through stroma lamellae. Okay, they are interconnected through stroma lamellae. Now, the space in between this inner and outer membrane is also known as periplastidial space. Now, within this stroma, if it is discussed, দেখো নেই হয়তো দরকার নেই বলে দেওয়া নেই, but still if it is discussed, then we'll find that this chloroplast is having their own DNA. They are having their own ribosome. So within the chloroplast DNA, ribosome, these are found, and even one most important is that within the stroma, a most important enzyme known as rubisco. This is very important. আমাদের এইটা রাখবে রুবিস্কো। This enzyme is generally found within this stroma. না, এখানে আমাদের যেটা আলাদা করে জেনে নিতে হয় এই জায়গায় সেটা হচ্ছে যে what is the function of this thylakoid and what is the function of this stroma, or in short basically chloroplast এর ফাংশন কি। সো বেসিক্যালি দেখো এটা আমরা জানি যে chloroplast, a chloroplast, they contain pigment known as chlorophyll. These are what? These are pigment which actually absorb sunlight. And as we have learned that sunlight is the major source of energy in photosynthesis. It's the ultimate source of energy in photosynthesis. We can simply say that that this chlorophyll, it absorbs sunlight and generally changes that light energy into chemical energy of food. That is how photosynthesis or synthesis of food occurs. So এটা আমরা জানি যে ক্লোরোপ্লাস্টের কাজ কি। হোয়াট ইজ দ্য ফাংশন অফ ক্লোরোপ্লাস্ট? ক্লোরোপ্লাস্ট দে হ্যাভ ক্লোরোফিল দ্যাট অ্যাকচুয়ালি হেল্পস ইন দ্য প্রসেস অফ ফটোসিনথেসিস বাই অ্যাবজর্বিং সানলাইট। কিন্তু এটা বুঝতে হবে যে বেসিক্যালি থাইলাকয়েড, অর ইউ ক্যান ইভেন সে ইন গ্রানা, আমরা ডিফারেন্সটা দেখে নিয়েছি, দ্য ডিফারেন্স বিটুইন থাইলাকয়েড এন্ড গ্রানা। তো দুটো বললেও কোনো অসুবিধা নেই। Thylakoid are in grana, or you can even simply say in the membrane of thylakoid. Will find different kinds of pigment. তাহলে কোথায় থাকে? মেমব্রেন থাকে। থাইলাকয়েড এর মেমব্রেনে কি থাকে? Different kinds of pigments. Now later on if we'll discuss, we'll find that different kinds of pigments involves, we can simply say different kinds of chlorophyll, different kind of chlorophyll. Okay, besides chlorophyll, carotenoids are found, and even xanthophylls are also found. These are also pigments. Now different kinds of chlorophyll includes chlorophyll A, chlorophyll B, and the most important one is chlorophyll A. Carotenoids are basically, they are orange pigments. And xanthophylls are basically yellow to orange pigment. Is it clear? Yes, sir. Okay. তো এবার যদি দেখি তাহলে এই ক্লোরোফিল বা এই পিগমেন্টস, তাদের কাজ কি? তাদের কাজ হচ্ছে টু অ্যাবজর্ব সানলাইট। তাদের ফাংশন কি? দে অ্যাবজর্ব সানলাইট। দেখো এখানে এটাও একটু বলে দেব যে লেটার অন ইফ উইল ডিসকাস, দেন we'll find that it is very surprising that photosynthesis is having two different phases. What are those phases? One is light phase, and the other one is basically dark phase. Other one is known as dark phase. It is also known as carbon reaction phase. Now we'll discuss later on why it is known as carbon reaction phase. Later on আমরা বুঝবো, আমরা পরে বুঝবো। আগে দেখি যে this light phase, it is a light dependent phase. dependent phase. Okay, light dependent phase. And basically দেখো এই টার্মটা খুব you can say that misleading, dark phase. তো ডার্ক ফেজ বললে অনেকে ভাবে যে ইট অকার্স ইন ডার্ক, কখনো ভাববে না। ডার্ক ফেজ যেটা বলতে চেয়েছে, আবার এটাও যদি বলি যে লাইট ইন্ডিপেন্ডেন্ট, তাহলে এটা ভুল হয়, বিকজ দিস ডার্ক ফেজ ইট ডিপেন্ডস অন লাইট ফেজ, ইট ডিপেন্ডস আপন লাইট ফেজ অবভিয়াসলি, এটা মাথায় রাখবে। দিস ডার্ক ফেজ ইট ডিপেন্ডস অন দিস লাইট ফেজ, এটাও মাথায় রাখবে। কিন্তু আমি বোঝার জন্য বলে দিচ্ছি যে এটাকে আমি লাইট ইন্ডিপেন্ডেন্ট ফেজ বলতে পারি, বাট নেভার সে দ্যাট। আমি এখন বোঝার জন্য বলছি, কি ঠিক আছে? বুঝতে পেরেছি আমি কি বলতে চাইছি? হ্যাঁ, লাইট ইন্ডিপেন্ডেন্ট ফেজ বলতে বুঝতে চাইছি ইট ইজ নট ডিপেন্ডেন্ট অন লাইট, but it is dependent on light phase. It depends upon the light phase. This phase occurs in the absence of light. It may occur, এইটা may occur in the absence of light even. Light, তার মানে এই নয় যে ইট ইজ নট ডিপেন্ডেন্ট, ওইটাই আমি বলতে চাইছি। এটা মাথায় রাখবো যে তার মানে এই নয় যে ইট ইজ, আমি লিখে দিচ্ছি, ডোন্ট থিঙ্ক দ্যাট ইট ইজ লাইট ইন্ডিপেন্ডেন্ট। আবার এখানেও বলে দিয়েছি, এটা মাথায় রেখো যে it depends, obviously it depends upon the light phase. সেটা আমরা বুঝবো, এই স্টেটমেন্টগুলো আমরা বুঝবো, কিন্তু জিনিসগুলোকে একটু মাথায় রাখা দরকার আছে যদি আমাকে বুঝতে হয়। It may occur in the absence of light. That is the reason why it is known as dark phase, because it is not dependent of, it is light independent. It don't depends on this light, but it is dependent on the light phase. ওই জন্য বলছি, don't think that this is light independent, but it depends upon the light phase. Is it clear? যা বলতে চাইছি বুঝতে পেরেছো?
হ্যাঁ স্যার।
আচ্ছা, generally because this statement is basically given in NCERT, ওই জন্য এটা বললাম। এই স্টেটমেন্টটা you can highlight, you can underline if when you find that don't think that basically it is independent of light. It is independent of light, obviously not. It depends upon light through light phase. লাইট ফেজের জন্য মানে লাইট ফেজে লাইট দরকার হয়, আর লাইট ফেজের উপর ডিপেন্ড করছে ডার্ক ফেজ। এনিওয়ে, যেটা বলার আছে, এত কিছু বলার কারণ হচ্ছে যে আমরা যদি দেখি তাহলে হোয়াট ইজ দ্য ফাংশন অফ দিস থাইলাকয়েড? বেসিক্যালি ইট অ্যাবজর্বস সানলাইট, and who absorb, মানে which molecules absorb sunlight? Basically the pigment molecules absorb sunlight. So basically we can say that within the thylakoid, this light phase, thylakoid, what is the function of thylakoid? Main function, the main function of thylakoid is that light phase takes place within the thylakoid. Light phase takes place within the thylakoid. Since in light phase we can see that it depends on light, basically we can say that the pigment molecules absorb light. So, and the pigment molecules are found within the membrane of the thylakoid. So basically the light phase generally takes place where? Within the thylakoid. And within the stroma, if you'll find, within the stroma, basically we'll find that the dark phase occurs. So এটা একটা কোশ্চেন, ইম্পর্টেন্ট কোশ্চেন আমাদেরকে জিজ্ঞেস করা হয় যে where light phase occurs within the chloroplast and where dark phase occurs within the chloroplast. Dark phase is also known as carbon reaction, and it is also known as carbon assimilation phase. Carbon assimilation phase. Okay. Acha. Reaction of photosynthesis. Chemical reaction of photosynthesis. chemical reaction is 6CO2 plus 12H2O in the presence of sunlight with the help of chlorophyll. What we see, C6H12O6 plus 6O2 plus 6H2O. Is it clear? এখানে তো বোঝানোর কিছু নেই, কিন্তু কিন্তু এখানে বোঝানোর কিছু নেই বললেও ভুল। বেসিক্যালি দিস কার্বন ডাইঅক্সাইড ইট গেট রিডিউসড, এইটা জেনে রাখো। This carbon dioxide is getting reduced. এটাই কোশ্চেন। বলতে পারো কেমিক্যাল রিঅ্যাকশনটা তো ছোটবেলা থেকে আমরা মুখস্থ করছি বা জানি, তো ওখানে আমাদের হয়তো কোনো প্রবলেম হয় না। এখন যেটা আমাদের আলাদা করে জানতে হচ্ছে যে দিস কার্বন ডাইঅক্সাইড গেটস রিডিউসড। ওকে, and we can say that this water it get oxidized. তো এখানে আগেই বলা হয়েছে যে ইটস আ অক্সিডেশন রিডাকশন রিঅ্যাকশন। আচ্ছা, এইখান থেকে আমি একটু বলবো, আমি আর আপাতত এখন বই অনুযায়ী এখন এগোবো না। বেসিক্যালি অক্সিডেশন অ্যাকর্ডিং টু ইউ is gaining of oxygen, isn't it?
Yes, sir.
We can even say removal of hydrogen. Can we say that?
Yes, sir.
Now can you tell me that if we can say that oxidation also means donation of electron?
Yes, sir.
Okay, so remember this thing first. And what reduction generally means?
Gaining of hydrogen and removing of oxygen and accepting electrons.
So reduction generally means gaining of hydrogen removal of oxygen and accepting ইলেকট্রন। তো এই জিনিসটা আমি প্রথমে মাথায় রাখবো। তাহলে এখানে একটা অক্সিডেশন রিডাকশন রিঅ্যাকশন হয়। উই নিড টু নো দিস ফার্স্ট। তো এখানে কি রিডিউস হচ্ছে? দিস কার্বন ডাইঅক্সাইড গেট রিডিউসড টু ফর্ম হোয়াট? বেসিক্যালি C6H12O6। এটাকে আমরা বলছি, হ্যাঁ, এটাকে আমরা কি বলি জানো তো? এটা কি বলি? কার্বন অ্যাসিমিলেশন। এই কার্বন অ্যাসিমিলেশনটা আমি বোঝাবো পরে, কেন কার্বন অ্যাসিমিলেশন বলছে আমি বোঝাবো। এটাকেই কার্বন রিঅ্যাকশন বলছে, এটাকে এই কার্বন রিঅ্যাকশন বলছে, জেনে রাখো। কার্বন রিঅ্যাকশন মানে কি? কার্বন অ্যাসিমিলেশন। ইজ ইট ক্লিয়ার নাও? কেন বলছে পরে আসছি।
Yes, sir.
In this process carbon dioxide get reduced to form glucose and this water is oxidized to form oxygen. আমরা প্রথমেই দেখেছি that source of oxygen in which is evolved in photosynthesis is basically what? Water. We have discussed earlier. এটা একটা প্রশ্ন আমরা পাই, what is the source of oxygen? The source of oxygen is water. The water get oxidized to form to release oxygen. Anyway, তাহলে কি বুঝলাম? একটা অক্সিডেশন রিডাকশন রিঅ্যাকশন where oxidation and reduction takes place, and this is what oxidation means and this is what basically reduction means. Now what we have discussed earlier is that this photosynthesis, it involves two different phases. One is light phase and the other one is known as dark phase. So what light phase actually means? It is dependent on light. In this phase light is generally absorbed. Light is the ultimate source of energy. Light is absorbed and we can say that the light energy is converted into chemical energy. Now, who helps in absorption of this light? Different pigments. So, different pigments, they absorb light energy, and what they generally does, they simply convert this light energy or solar energy into what? Chemical energy. Now, what this chemical energy actually is, we'll discuss that. Now, in dark phase, what happens? দেখো আমি আগে কনসেপ্টটা দিই। লাইট, আচ্ছা লাইট ফেজে কি হয়? এই যে কেমিক্যাল এনার্জি বলছি, so basically during light phase this light energy is converted to chemical energy. What this chemical energy actually is? Chemical energy of ATP. So এই ফেজে কি হয়? এনার্জি তৈরি হয়, এনার্জি is generated. Is it clear?
Yes sir.
দেখো আমরা যদি একটু ভেবে দেখি, প্রথমে ওই জন্য এইটা বলা আছে, ইটস ভেরি সিম্পল, আমি জাস্ট ট্রাইং টু ডিসকাস উইথ ইউ, ইটস ভেরি সিম্পল। প্রথমে এটা বলা ছিল যে বেসিক্যালি অল অ্যানিমালস ডিরেক্টলি অর ইনডিরেক্টলি দে ডিপেন্ডস অন প্ল্যান্টস ফর দেয়ার ফুড। তো ভেবে দেখো অ্যানিমালসও তো উই ক্যান, আমরা তো লাইট এনার্জি ইউজ করতে পারতাম, উই ক্যান ইউজ আর উই ক্যান ইভেন অ্যাবজর্ব অ্যান্ড উই ক্যান ইউজ দ্যাট লাইট এনার্জি টু সিনথেসাইজ আওয়ার ওন ফুড, বাট পারি না। We can't. Only plant can do that because plant have that pigment that helps in absorption of light. So what they does, with the help of those pigment, they absorb light and they convert into chemical energy. এবার আমরা কি, আমরা যদি করতে পারতাম তাহলে তো হয়ে যেত, কারণ আমাদের বডিতেও তো এটিপি তৈরি হয়। এনার্জি, এটিপি ইজ দ্য এনার্জি কারেন্সি ইন অল লিভিং অর্গানিজম। তো আমাদের বডিতে এটিপি তৈরি হয়, বাট উই কান্ট ইউটিলাইজ লাইট এনার্জি অ্যান্ড কনভার্ট দেম ইনটু এটিপি। So now you understood যে actually light phase কি হচ্ছে, light phase এনার্জিটাকে জেনারেট করছে কি? ATP, because ATP can only be utilized within the living organism. This is the energy currency, isn't it? Besides ATP, even we'll find that Na, DPH, later on আমি আসছি। তো আপাতত তুমি জানো যে এনার্জি জেনারেট করছে, and besides এনার্জি আর কি পাচ্ছি? NADPH ও পাচ্ছি, এটা জেনারেট হচ্ছে। তো হোয়াট ইজ দ্য রোল অফ NADPH, দ্যাট উই আর গোইং টু ডিসকাস লেটার অন, ঠিক আছে, আমরা পরে ডিসকাস করছি। কিন্তু আপাতত এইটুকুই বুঝবো যে দিস লাইট এনার্জি ইজ কনভার্টেড ইনটু হোয়াট? ইট ইজ কনভার্টেড ইনটু ATP, মিন্স এই ফেজে কি তৈরি হচ্ছে? এনার্জি, অ্যান্ড দ্যাট এনার্জি generated during light phase is utilized to fix or assimilate carbon dioxide into C6H12O6. বুঝবে? আশা করছি আমি যেভাবে বলে দিয়েছি এমনি বোঝা যাচ্ছে যে এখানে আমি এনার্জিটা তৈরি করছি, আর এখানে আমি কি করছি, ওই কার্বন ডাইঅক্সাইডকে কিভাবে আমি ইউজ করে গ্লুকোজ তৈরি করব, তার জন্য তো এনার্জি লাগবে, বিকজ ইটস আ অ্যানাবলিক রিঅ্যাকশন, ইজন্ট ইট? সিন্স photosynthesis is what? Photosynthesis is actually an anabolic reaction. Now, anabolic reaction, basically in anabolic reaction, energy is utilized. So, এই এনার্জিটা কোথায় থেকে পাচ্ছি? Energy is utilized. So, where from this energy is coming? The ultimate source of energy is the sun, which is converted into chemical energy of ATP, and this ATP is utilized to prepare food. Is it clear? বোঝা গেছে? Now, can you tell me, লাইট এনার্জিকে, খুব ভালো কথা, ক্লোরোফিল অর পিগমেন্ট, ডিফারেন্ট পিগমেন্টস কনভার্ট করছে এটিপিতে, কিন্তু একটু মাথায় রাখবো যে the process by which ATP is formed is known as phosphorylation. It is known as phosphorylation. What it is known as? কি বলে বাবা? Phosphorylation. Now what phosphorylation actually is? Adenosine diphosphate, when it is combined with inorganic phosphate, it forms adenosine triphosphate. So this is what actually phosphorylation is. Now why it is said to be phosphorylation? একে কেন বলছি phosphorylation? কারণ এখানে what? Phosphate is added. Phosphate is combined. That is why it is known as phosphorylation. কারণ লাইট রিঅ্যাকশন ডিসকাস করতে গেলেই আমার যেটা আসবে সেটা হচ্ছে সাইক্লিক ফসফোরিলেশন আর নন সাইক্লিক। ইয়েস, তো আমি এতদিন হয়তো দ্যাট রিলেশনশিপ, ওই যে সম্পর্কটা হয়তো এতদিন একটু অসুবিধা ছিল। এখন তুমি নিশ্চয়ই বুঝতে পারছো যে আমাকে যদি এই লাইট রিঅ্যাকশনটা বুঝতে হবে, দেন ফার্স্ট আই নিড টু আন্ডারস্ট্যান্ড হোয়াট ফসফোরিলেশন ইজ, বিকজ লাইট ফেজে আমরা টু টাইপস অফ ফসফোরিলেশন পাই, সাইক্লিক অ্যান্ড নন সাইক্লিক। তো লাইট ফেজে কি হচ্ছে, সাইক্লিক অ্যান্ড নন সাইক্লিক ফসফোরিলেশনের মাধ্যমে কি তৈরি হচ্ছে বলো, এবার তুমি বলো। এটিপি তৈরি হচ্ছে তো, এটাই তো হচ্ছে।